is that basically there are specific organisms in genetics and in research that I like for you to be familiar with, because we encounter these kinds of organisms when we're working genetics problems. So, looking at the sporophyte, you know what the word phyte means? Whenever you see that word, you know you're talking about a plant. That's what that means, sporophyte. And it's diploid. It undergoes meiosis and forms haploid spores. And then this will undergo mitotic division and form a gametophyte. But we talked about this in detail. This is a shorter version of it as we had gone through it. And the gametophyte, which is a multicellular haploid structure actually, and it will form gametes and then fertilization, forming the zygote, which is diploid, mitosis, which will make the adult organism. In yeast, we have a genetic series, a lab dealing with yeast. What I want you to know from this is that the mating types in yeast are alpha and A. So what are the mating types in humans? Anybody? I'll give you a hint. One of them is male. Male and females are the mating types in humans. This is the alpha mating type. It will mate with an A and form a zygote. I ask such hard questions. So this, in yeast, the cells, they really can exist as haploids and they can exist as diploid. But two alphas cannot mate and two A's cannot mate. Only opposite mating types can mate and form a zygote that's diploid. And then this zygote will undergo meiosis, 1 and 2, and form, again, 4 haploid cells. And so of all the combinations that you can make of big B, little b, big D, little d, that will undergo meiosis and form 4 haploid cells. And these would be 2 alphas and 2 A's. And those will be the mating types. So in yeast, they start out as haploid. They form a zygote that's diploid, undergo meiosis, and form haploid cells again. Unlike humans, yeast is different from humans. We start out as diploids. Our gametes are haploid, and they fuse to make a diploid organism. So these are, yeast is different. And so all of these haploids. So why have meiosis in yeast? Because yeast can reproduce by budding, and it can be reproduced by sexual reproduction. So yeast have asexual, which means that it can reproduce by budding, or sexual, that's the mating types. So why would yeast, why have a mating types and sexual ways of reproduction? What good is it? What significance do you think it is?
They exchange DNA, so they don't have as many genetic.
Actually, that's true. It increases the variability in the population. It increases the variability in the population because before the yeast was little b, little d, and if they reproduce, they will produce only little b's and little d's. So with this one, big D and big D, by forming the zygote, we form 4 different. So it increases the variability.
It increases the variability in the population, but actually they just mix genes, they mix DNA.
Because of recombination, right?
Right.
So yeast has 17 chromosomes. So the zygote will have 34 chromosomes that mix, recombine, and move through meiosis. So this is an oversimplified scenario, but it does, I wanted you to understand this because we have a very long lab dealing with yeast. I would like for you to go back to these notes that we say we're talking about yeast because it may be helpful in answering some of the questions. Okay? This is Neurospora. Very similar to yeast, but it has a mating type plus and a mating type minus. Just like yeast, Neurospora is also a fungus.
What?
Fungus, right. You knew that though, right? Yeah, it's a fungi. So we have a mating type plus, mating type minus. They fuse and form a diploid zygote, undergo meiosis 1 and 2, and then each one of these will undergo another mitotic division. So notice we have 2 little b's, little d's, 2 big b's, and 2 little d's, and so on. So it goes from an 4 spore to an 8 spore. And that's the difference in Neurospora. When we talk about recombination and gene conversion, we'll come back to this and explain how we know these things can happen. Yes?
Why do you need to increase the variability of yeast?
Say again.
Why do you need to increase the variability of yeast?
In all sexual reproduction, reproducing organisms. So because of sexual reproduction, we increase the variability in our offspring. And the reason why is because the more variable your population is, the more likely they will survive environmental change. You follow? If everybody was little b, little d, if the environment changed, they all die. You follow? Am I answering your question, or are you asking something else? Yeah, but it's true for any organism. That's the only reason. Did you have something else that? Okay. So, but you know, we're not in genetics. I should know other parts of the life cycle, but really in genetics, all I'm really concerned with is what happens to the chromosomes, and that's why I'm limiting it to the nuclear divisions of what happens after the division, because in genetics, I can use certain organisms to explain specific questions. If I want to find out more, get more information on a particular question, the choice of organism that you use is very important. Chlamydomonas is a green algae. It's a single cell green algae. And it has 2 mating types as well. The 2 haploid cells can merge to form a diploid zygote, as shown here. And so these are the different kinds of nuclei that we have formed. The reason for the studying Chlamydomonas, if I want to study photosynthesis, it is an algae. And as well as the genetics, what does the genes have to do with chlorophyll or whatever the questions may be? You would choose an organism like this to study the genetics as well as, say, algae, or can imply what happens in plants. Chlamydomonas is all 3 of these were haploid organisms, but they basically reproduce the same way. Haploids fuse to form a zygote, and the zygote undergoes meiosis. I used to use this one a long time ago to explain plants. And there's no easy way to go through it, but you have a better drawing of this when we did gametogenesis in plants. Matter of fact, we had talked about endosperm, double fertilization, and that's all this does. And this is, of course, showing the stages of meiosis and what happens as we go through the different divisions of pairing and so on, when the centromere divides and continuum. So I think you know this pretty well, that the mouse is 2N only because of the fusion of haploid germ cells. That shows the female forming the polar nuclei